As we alluded to when we looked at the structure of DNA, part of what is so incredible about it is how the structure actually helps DNA replicate or copy itself. And that's real important because if cells are going to reproduce and they need to pass on all of their genetic information, you have to make copies of it. So for starters, one of the things that happens here, we talked about how all of these molecules in one of the strands are bonded covalently, so they're not going to come apart. But in the middle here, we've got hydrogen bonds that are a little bit weaker. And it starts to get unzipped in different spots in the DNA. It's not always the same spot. And then you've got nucleotides that get added on, which are these little dots along the way there. And this zipping gets bigger and bigger until it's totally unzipped and everything's been copied and the duplicate strands are joined together, and now you've got two strands of DNA, with this end being a copy of that side and this end being a copy of that side. How does it work? There's some enzymes that help do it. Enzymes are proteins, if you remember. Enzymes typically end in ASE, which makes it a little bit easier. So the first enzyme that does its thing is DNA helicase, and that's what goes through and unzips these hydrogen bonds. The next thing is DNA ligase starts putting on the nucleotides on the side where it's been unzipped. Now, how does it know what nucleotide to put there? Well, if you remember, anytime there's a G, it knows a C has to go on the other side. Anytime there's an A, it knows there has to be a T on the other side. Anytime there's a T, it knows there has to be an A on the other side. And anytime there's a C, it knows there has to be a G on the other side. So ligase is able to link up these nucleotides so that you end up here now with a complete strand there and a complete strand there after it's been unzipped. Now the last part that has to come along is these two strands must be rejoined back together in hydrogen bonds, and that is the purpose of DNA ligase.